Good morning or afternoon. Hi, this is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. This webinar is part of a series, as you know, if you've attended these webinars before, sponsored by the ASCCT and the European Society for Toxicology in vitro together. I would encourage you to visit both of our websites and consider becoming a member to support the programs that both societies undertake, like this webinar series and other activities. We host regular meetings and pursue other activities to promote computational and in vitro toxicology. And we also offer training and education and support of early career scientists and students in their endeavors. This webinar is recorded and the recording will be posted on the ASCCT website a few days from now. Also, your lines are muted. If you would like to ask a question, you are encouraged to do so. Please enter your question in the Q&A feature in the toolbar at the bottom of your screen there. The main Q&A will be after both speakers have finished. And I would also encourage you to say hello in the chat and let us know where you're joining from. So I just wanted to mention that ESTIV has a Congress coming up. Their call for abstracts and registration is open. It's going to be in November this year in Barcelona. So feel free to go to their website and check that out. We have a number of upcoming webinars through April. We're going to be featuring some students and early career scientists who've won awards from both societies in the past last year, and also some top vocal experts kind of mixed in there. So I encourage you to keep up to date or join our email list to find out when each webinar is scheduled. Next month, we have a webinar on March 17th. You can register now on the website. It is a mix of 3 presentations talking about advanced models for skin drug testing and disease modeling, and lung disease modeling, and also an in vitro hepatopulmonary co-culture. So that should be very interesting. Hope you'll join us. And for today, we have a feature of growing cells in serum-free media for toxicity testing. Our first presenter is going to be Barbara Jozef from the Swiss Federal Institute of Aquatic Science and Technology. Dr. Jozef earned her PhD degree in biochemistry from Belgrade University in 2018 on the topic of colored xenobiotic degradation, with a special emphasis on the eco-genotoxicological impact of the method developed. She's currently working on a project that aims to develop serum-free project for fish shells. That's what she's going to be talking about today. The goal of this research to help understand the nutritional requirements of fish shells and help advance alternatives to fish replacement. Barbara is also an award winner. She won the ESTIV Best Poster Award at the 11th World Congress on Alternatives to Animal Use in the Life Sciences last August. And this award is presented to a PhD student or early career scientist. And winners were selected based on scientific excellence, innovation, and clarity of the poster presentation. For this award, she wins a €500 prize, an ESTIV membership, and her research is going to be highlighted in a special edition webinar today, and she'll have an interview published in web and social media accounts of ESTIV. So if you're interested in being recognized, keep track of ESTIV and ASCCT in the next coming year. We offer, both societies offer a lot of opportunities to do that. I'm going to introduce our second speaker now because we'll just roll into both of the presentations. Dr. Aline Chari is R&T Associate at the Luxembourg Institute of Science and Technology. During her PhD thesis with LIST and the University of Trier, she worked on the development of an in vitro co-culture model to study respiratory sensitization. This model was patented in 2017 and recently submitted for validation to ECFAM. Aline defended her PhD thesis this year and was awarded in 2018 the Lush Prize, Young Researcher. And she's working with PETA on the replacement of animal-derived components for cell culture application. So I will stop sharing and turn it over to Barbara.
Thank you, Chrissy, for this nice introduction and for organizing this. Allow me 1 second to start sharing my screen. I hope that you can now see.
Yep.
So it is, yeah, okay, I can start, right? It is great opportunity for being here today. Thank you for joining. Hello from Switzerland. I'm so excited to be here today talking about my serum-free media development journey. Also, this is a great opportunity to share my fresh passion for fish cells. Now, you probably wonder what is there exciting about a fish, let alone a fish cell. Well, let me elaborate on that, as well as to show you what it has to do with going serum-free. The man-made chemicals have become an essential commodity of our everyday lives. They are produced by industry facilities around the world. Regulators trying to minimize the impact of environmental damage, they are making companies conduct safety testing on such chemicals. Yes, you guessed it. Mainly, this testing is done on fish. You also may think, well, it's just a fish, but if I tell you that we use millions of fish on a yearly basis solely for this purpose. In order to replace usage of fish in such tests, the research group led by Professor Christian Schimmer is working on animal-free alternatives. Now, the permanent fish cell line of a rainbow trout, which is a key species for environmental protection, is used for such endeavor. The most advanced procedure is the RT-gill WE1 cell line acute toxicity assay, which can predict fish acute toxicity. It has been accepted as an ISO standard and as well as an OECD test guideline. However, while these advances paved the way to replace the fish, the routine culturing of fish cells still requires fetal bovine serum, an ill-defined mixture that allows cells to proliferate in the culture. Apart from that, FPS is derived by highly unethical method, and furthermore, there are several scientific and technical problems with regards to use of FPS. Therefore, our goal was to move not only to fish, but rather to completely animal-free testing. And the final step would be to eliminate the need for the FPS. Our serum-free project has several goals. First was to identify which ready available supplements might substitute for FPS in theory. Next, we developed a short-term cell proliferation assay for a quick evaluation of these supplements. And this goes hand in hand with the design of experiment that points to a specific component that might be crucial and to help us understand how these components might interact. Altogether, this drives towards the media formulation to our desired end goal to do long-term culturing without the use of FPS. How complex can it be to replace one universal-like component, you may ask yourself? Well, since the FPS is highly complex and it has been reported that there are more than 1,000 compounds presented in there, I would dare to say it's not that simple. However, separation of these components by their function and/or structure can give us a better overview and offer some guidelines when it comes to the FPS replacement studies. Also, the cell culture media pyramid, as proposed by Van der Valk and the co-author, is a very helpful tool as it nicely summarizes these categories and also emphasize the need for cell specificity when it comes to the media development. Altogether, this provided us with a very good starting point since we decided to build our media from scratch, meaning we used only Leibovitz 15 as a starting base. And this is a very well known and validated basal medium for fish cells. Furthermore, our experimental design allowed us to eliminate few factors right at the beginning phase, such as adhesion factors and trypsin inhibitors, and it made our endeavor more achievable, and I will speak about it in more details in the coming slides. So we urged to rationalize this undertaking even further. So we employed a sequential approach that consisted of 4 phases, where the phase 0 was actually to develop a cell proliferation assay, which was followed by the phase 1, where we actually built our basal serum-free media, and after that we would tackle cell proliferation. Finally, we would validate our serum-free mixture by scaling up and doing long-term culturing. So in order to be able to follow the desired outcome, the first focus was actually to establish a high throughput assay that would support rapid identification of the relevant FBS replacement components and to accommodate assessment of several endpoints relatively fast and reliable within the minimum numbers of days. So on the first day, or we call it day 0, is when we seeded the cells in 96 well plates under the FBS conditions. We left the cells for 24 hours so that the cells could properly attach. This was very crucial because the gill cells are anchorage dependent, and there are factors in FBS which allow them to attach. Without this, they are unable to function properly. Afterwards, the cells were rinsed and the media was exchanged to a serum-free mixture of various components, either individually or in the mixture, that were done in L15. And then we followed the outcomes until day 7, where we actually measured cell viability, did the cell counting, and followed the cell morphology. So we used the developed assay to first observe what is happening to the cells when they are in unsupplemented media. We saw that the gill cells, which were only in L15, presented here as circles, do stay viable for days, but at the same level. Considering that the cells in FBS media presented here as square, had shown a significant increase in cell viability. We argued that this gap was opening due to the differences in the cell proliferation, and by performing direct whole well cell counting, this was confirmed, and we could nicely quantify the difference between the 2 conditions. Also, this gave us wide enough experimental space to start building our media. So at phase 1, it was important to start investigating nurturing supplements that would build our basal medium. From the graph here presented, we can see the impact of the selected supplements on the gill cells. The results are shown as a percentage of cell metabolic activity normalized to the cells that were in unsupplemented media, or L15. You may notice there are 2 benchmarks on the graph. First is the gray dotted line, which presents the minimum requirements, meaning that if some cells had outcome below this threshold, that there was probably some sort of toxicity happening. The other benchmark, the violet dotted line, was actually our goal line or threshold to be reached, and it was done based on the cells which were exposed in FBS media, so-called positive control. Interesting thing is that certain mixtures of supplements are showing better outcomes and are getting quite close to our desired goal, which is the FBS cells. This leads me directly to my next point. So from the cell biological perspective, we can say that the cells need certain factors presented here as A, B, and C in order to survive or to maintain viability. Whereas completely different additional sets of factors is needed for them to grow in total mass and volume, which will be accompanied by a proportional increase in numbers of cells. Term proliferation would be maybe more suitable, and it is driven mainly by the group of supplements listed here. Now, taking a step back and observing a bigger picture, as the targeted cell lines are epithelial in nature, we could then comprehend the basics of parameters that control cell growth, proliferation, and even differentiation of cultured cells in vitro. Now let's take a look how this screening of crucial supplement went in our case. So the bar indicate here the cell proliferation of a gill cells in a serum-free mixture and are again normalized to the cells in L15 alone or no cell proliferation. The new supplements tested were added to the previously best mixture, either individually or in combination. Significant thing about this phase is that for the first time, we can observe an overperformance of our cells in serum-free mixtures when compared to the FBS cells. As mentioned before, we were also following the cell morphology, and here you can see the representative phase contrast images of the cells that were cultured in serum-free mixture compared to the cells in FBS. So I hope that you can appreciate from your screens that the morphology of these cells is quite comparable. With this strong enough data set, we were encouraged to move to the next phase in our project, and that was to scale up our experimental system by culturing cells in a flask. Now, this is where we tested different adaptation process. The first one was following the sequential approach, meaning gradual reduction of FBS with increasement of serum-free media by each media exchange. Basically, we seeded the cells in FBS, and after 3 days, we start to wean them from the FBS. Hence, we named these cells weaned. You can see how the gill cells looked during this time. So by the day 20, we have reached 0 FBS mark, which meant that we were prepared to go for our first passaging. And we did so by using only serum-free media. We followed the cells and realized that not only cells were attaching, but they were also starting to proliferate. Illustrated by a timeline here, we had this first hurdle tackled, which was the first passage. The cells were ready to be passaged again. So we did so, and then we were approaching very important benchmark set up by our lab, which is the passage number 10. Since previous successful experience in trying to adapt the gill cells to commercially available serum-free media failed at this point, we were very eager to see how the things will evolve this time. So not only did we reach this passage number 10, but the cells were really doing great. I hope that we can agree that reaching this benchmark is rather high goal to achieve for any type of mammalian cells when we are trying to go serum-free. Furthermore, our cells are currently in passage 15, and we are looking forward to exploring their passaging limit. Now, I was keen to pushing the timeline for adaptation process to be shorter. So we tried the direct adaptation. In this case, we followed the pipeline of previously developed short-term proliferation assay. So we seeded the cells in a flask in FBS, allowed them 24 hours to attach, and then we exchanged the media to 100% serum-free. We left cells for a few days, we monitored them and decided to try to passage them for the first time. We did this and the cells were catching up, so we passaged them again. And then with occasional clumps and a bit of a slower growth pace, these cells hang in there and made it to currently passage 11, again surpassing the passage number 10 benchmark. Next hurdle to tackle for us was possibility of a long storage of these cells, as this would mean safe preservation for the longer run. As our serum-free mixture already consisted of several different components and was showing good results, we decided to go with a protocol that we would use as if we were using the FBS. So we froze the cells using 90% serum-free media and 10% DMSO. We left them in the freezer and then we thawed them. And this is how the cells looked 24 hours after thawing. I hope that you can see that there was, right from the start, signs of healthy attachment that was followed by cells catching up, and we were able to culture them from then without major problems. So where do we go from here? I'd like to highlight that we are keen on elaborating this project further. We are looking into fine-tuning of our serum-free mixtures, as well as to assess the performance of serum-free cells to known toxicants. Moreover, we would be very happy to explore the application of this media to other types of rainbow trout cells, lines, or even fish cells in general. Last but not least, I would like to state that we are committed to making our serum-free mixture publicly available. We believe this is a way forward that will benefit not only scientific community, but far beyond. Since at this very moment, I am unable to disclose the exact supplements we used as publication process is underway, I would still like to provide some more insight. So our scientific journey started from a serum-containing media, which we used for routine culturing of these cells, and we were able to move to a serum-free media, which means that we don't use FBS anymore, but there are some components that are of animal or human nature. So our current serum-free media consists of 27 supplements on top of L15. Currently, 2 of them are still categorized as animal origin. Based on the figures that we have, I'm certain that reaching our final overarching goal of going animal-free as well as chemically defined is achievable. So before I thank you for your attention, allow me to express my sincere gratitude to Professor Dr. Christine Schimmer for mentoring me and supporting me throughout this project. I would like to thank to Dr. Hans Michael for his selfless sharing of design of experiments knowledge. Melanie, my colleague, for always having my back as she is our main cell technician. To Frank, my master student, for helping me advance cell proliferation assay. Furthermore, I am grateful for Swiss 3LCC for funding my project and Environmental Toxicology Department at EAWAG. So I thank you for your attention, and I hope you have gained some insights into why our fish great model. And also, I would like to finish with this slide, which nicely says it with a little bit of math, that with fetal bovine serum from 1 cow, we can do 151 acute toxicity assay that could actually help save 6,384 fish. But after all, shouldn't it not be that every fish and every cow counts? So thank you.
Wonderful. Thank you, Dr. Jozef. We are going to move on to the next presenter, and then we'll handle all the questions together. So thanks. Keep putting your questions in the Q&A, and we'll turn the floor over to Dr. Shari.
So normally you are able to see my screen. Good morning and good afternoon, everyone. So today I will present you my work on the replacement of the FBS in cell culture application with a concrete example using the A549 cell line. So to give you some background on the L549 cell line, it's an immortalized human epithelial alveolar cell line. And these cells are commonly used as a model of alveolar type 2 epithelial cells in respiratory research and testing. They express intracellular multilamellar bodies, and they produce surfactant, which allows their culture and exposure at their liquid interface, or ALI. And finally, the A549 cells are relatively inexpensive to grow and easy to grow. So it's possible to test a large number of chemicals. So about their cultivation, so according to a supplier instruction, the A549 cells should be cultivated in basal medium, so DMEM for our case, supplemented with 10% of FBS. So as already described in Barbara's presentation, FBS contain more than 1,000 components, including protein, hormone, and undefined constituents. And the use of FBS raises scientific, legal, and ethical issues. So the use of FBS is indeed known to influence the cellular phenotype and the gene expression, which can affect the outcome of a study. But also it may be a source of variation, as this could be really problematic, especially when you want to, for example, to reach the validation of an in vitro model. So this variation in composition was recently demonstrated to contribute to inter-laboratory variability of cell growth and behavior of A549 cells in a recent study from 2021. So how do we do to replace FBS in the media? So of course, it's possible to develop your own media, as Barbara done, but it's quite time-consuming. So for this study, we decided to choose 4 different FBS-free media, which are commercially available. And in addition, we use this commercially available medium as it's accessible for every researcher. So for the 4 media that we selected for the study, there is CNTPRA, which is a complete medium, which is provided by Cell&Tech, and it's a chemically defined medium. Then we have the ex vivo medium, which is provided by Lonza. So it's also a complete medium. And this one is xeno-free. So it means that it does not contain any animal component, but it contains human components, such as a human serum albumin. But also depending on the lot and the type that you choose, it can contain human transferrin or recombinant transferrin. Then you have the HL1 medium from Lonza. So this one is a supplement to add in DMEM. It's a serum-free media, meaning that it contains animal components such as bovine catalase. So for us, it was not the best choice because we would like to avoid any use of animal supplement in our method. And finally, we have the serum-free XF212 provided by TNC Bio, and it's a supplement to add also in DMEM, and this one is chemically defined. So now we have the 4 different media, so we need to adapt our cells. How do we do? So this table was presented in a review paper that Barbara also shown in our presentation. And here we have 4 different possible strategies to replace the FBS. So the reduction of content serum, where the cells are placed in serum-free medium with a percentage of FBS, and then we decrease the percentage of FBS until reaching the 100% of FBS-free medium. Then we have the sequential adaptation, where the cells are first cultivated in normal medium containing 10% FBS. And then from each passage, we decrease the percentage of normal medium and we increase the percentage of serum-free medium until again reaching the 100% condition of serum-free medium. Then we have a third possibility where the cells are adapted like sequential adaptation, but with conditioned medium. And finally, the inside adaptation, which is a kind of direct adaptation of the cells to the serum-free media. So for our study, we choose the sequential adaptation and the direct replacement. And we defined the 6 dilution before FBS was completely removed for the sequential adaptation. So first, for the direct adaptation, it failed for all our media. And finally, for the sequential adaptation, it was successful for two media, the ex vivo and the CNT-PRA media. We have not assessed if quicker transition with less dilution, but it could be successful also. And it's important to say that the supplier of XF212 mentioned that depending on the cell type, we could need to coat the culture substrate or to add some recombinant growth factor or mods. But in this study, we used uncoated cell culture substrate, and we didn't optimize any of the cell culture media. So in our study, the two selected FBS-free media are the CNT-PRA and the ex vivo, where the characteristics of the medium are repeated in this table. And we kept, once the cells were adapted, we keep the cells in FBS-free media for at least 3 passages. So this doesn't mean that the cells were not able to grow after 3 passages, but we wanted to freeze them before to use them for other experiments. But of course, you can keep the cells at least 20 passages as you would do for the regular cells grown in FBS. So now about the cryopreservation, it could be also problematic in the absence of FBS. So we would also try different protocols. So different conditions were tested to evaluate how the cells should be cryopreserved in the FBS-free media. So we have here the classical cryopreservation in medium plus 10% of DMSO. We use also the CNT Cryo 50 and ProFreeze, which are 2 animal-free freezing medium. And finally, we use also medium with DMSO and human serum albumin. So to determine which freezing medium should be used, the cells were counted directly after thawing at day 0 and then seeded in 6 well plates in their respective media for 1 week. After that, the cells were detached and counted, and the number of cells at day 0 and day 7 were compared to evaluate the growth 1 week after thawing. So here the star indicates a significant difference between the different freezing media, and the circle indicates the difference between day 0 and day 7. So for CNT-PRA, we can see a decrease in cell number when the cells were cryopreserved using human serum albumin. And for ex vivo, human serum albumin and ProFreeze show an increase in cell number after 7 days. So of the tested option, the ProFreeze freezing medium was chosen to cryopreserve our cells. So now we can focus on the morphology of the cells, switch to FBS media. So you can see that ex vivo and DMEM show similar morphology with cuboidal cells. And the CNT-PRA, we can see that we have a kind of mix of cells with different morphologies. So we can see small and cuboidal cells as DMEM and ex vivo cells, but also very, very large cells. Then the doubling time of the cells was calculated for each media. So the cells were counted every day for 2 weeks. And based on this growth curve, the doubling time was determined with the 21 hours for DMEM cells, 51 hours for the CNT-PRA, and 33 hours for the cells grown in ex vivo media. So the doubling time are greater for cells grown in FBS-free media, and more especially for the CNT-PRA media. So now it's important to evaluate the functionality of the cells and to verify that cells grown in FBS-free media are still reacting as they should. So we saw in the introduction that an important function of alveolar type 2 epithelial cells is to secrete surfactant. So the DMPO test was conducted to verify the presence of surfactant, and a drop of DMPO is put on the cells cultivated at their liquid interface. And a small diameter of the droplet indicates a lower surface tension and the presence of surfactant, while a large diameter of the droplet indicates a high surface tension. So all the media resulted in cells with similar surface tension, and thus some surfactant was secreted using all media. So still at their liquid interface, the gene expression was assessed. So we selected genes relevant for alveolar type 1, type 2, and secretory cell markers. So the results demonstrate a drastic increase for alveolar type 1 and type 2 gene expression for cells transition to a CNT-PRA medium, which may indicate the presence of type 1 and type 2 cells. And this can confirm the mix of cells that we observe with a microscopic observation. So the same was done in a submerged condition, and we observed the same results as for the algae. Now, finally, the cells were stained with a lithotracker, here in gray on the picture, and this stains the acidic compartment, and it is used as a marker for multilamellar bodies that are typical for type 2 alveolar cells. And interestingly, as you can see, the very large cells found in A549 cells culture in CNT-PRA medium were negative for multilamellar bodies. And in addition, the multilamellar bodies were relatively larger in cell culture in CNT-PRA medium. So again, these results may indicate the presence of type 1 and type 2 cells in FBS-free media, the CNT-PRA. So in addition to the functionality assays that we performed, we also have done the performance assessment of the cells. So first, A549 cells were seeded in a submerged condition and exposed for 24 hours to different concentrations of SDS. So we can see here that the dose response curve of DMM and ex vivo are quite similar, while the cells transition to a CNT-PRA are more sensitive. And then we calculate the EC50 value, and indeed it's 4 times lower in CNT-PRA as compared to DMM cells. Finally, A549 cells were exposed to LPS for 24 hours, also in submerged condition, in order to compare the cytokine release. And once again, we can say that CNT-PRA are reacting differently, and they are releasing more cytokines as compared to the other media. And for example, the interleukin 18 is very interesting here, as it is mainly secreted by type 2 cells, and there is a basal secretion of the cytokine only for this medium. So this really reinforces the hypothesis that we have more type 2-like cells in CNT-PRA. So to conclude, the A549 cells were successfully transitioned to ex vivo and CNT-PRA medium using a sequential process. Ex vivo and CNT-PRA are viable alternative to FBS supplemented medium. So in one end, the ex vivo culture cells retain the characteristic and functionalities of cells culture in DMM plus 10% FBS. And in another end, the CNT-PRA culture cells have different morphology. And we could explore the differentiation into type 1 and type 2 alveolar epithelial cells. So we really need to choose the medium depending on the study. So as next step, we already submitted our results for publication, and we aim at testing the transition cells in a different lab. So please do not hesitate to send me an email if you are interested to get our cells. So finally, that's all for me. So first, I would like to thank you for your attention. And of course, I would like to thank Liz. And more especially, I would like to thank my colleague from the Environmental Health Group who participated to this work. In addition, I would like to thank people from PETA who participated to this study. So thank you.
Thank you, Dr. Shari, for that nice presentation. Both of you have really given a lot of really excellent information to people who are hoping to transition their cells as well. So thanks to both of you. We have a lot of questions in the Q&A, and it looks like, Barbara, you have been busy answering some of them already. So if attendees, if you want to take a look at some of the answered questions, you can see some of the answers there. And then we'll see as more questions come in, how the discussion goes. Okay, so well, maybe we'll start with Barbara. One of the questions that looks like you were starting to answer, it's from Chander Nagy about serum starvation potentially inducing autophagy in the cells that you're using. Wondering what you have to say about that.
Yes, so thank you for this interesting question. Definitely, we do know that the cells which were only in L15, where there was no FBS, were looking rather different in their morphology, and they were just not doing great. I would argue that this is for surely not happening in our serum-free mixtures, since the cells proliferate so well, and they are doing so from a few months ago. In terms of doing some comparison, we have a lot of things underway, and this 96 well plate cell proliferation assay is kind of our first measurement of the performance of the cells in comparison to the FBS cells, and especially when the cells are starved, meaning when they're only in L15.
We'll take a question for Aline. Did you check any EMT-related gene upregulation, such as vimentin or E-cadherin?
We just checked the genes that were presented in the table, so only secretory marker, alveolar type 1 and type 2 epithelial cells.
Okay, another question for Barbara. Did you notice a change in the doubling time with serum-free media?
So we are currently working on this. I would say that they're not on point as the FBS cells, but rather close, if I may eyeball it at this moment. But definitely this is the first thing that we are looking forward to actually quantifying. What is also interesting that I didn't mention is that these fish, these cells come from cold fishes. So basically their doubling time is a bit higher than the actual mammalian cells. So they're a bit slower to grow comparison to the maybe cells that Aline is using.
Now, Aline, if any of these questions are relevant to you and you want to jump in with your experience, please feel free to do so. Okay, so I'm going to dig into one question that people asked quite a lot, which you answered a little bit, but was about metabolic activation. And so I'm wondering for both of you, did you check to see if there were any differences in the serum-free media grown cells in metabolic activation compared to your original cells?
Go ahead, Aline.
Sorry, I had internet issues. I understand only your part of the question.
Oh, some people were asking about metabolic activation and whether there were differences between the original cell populations and the cell populations in serum-free media.
For me, regarding the metabolic activities, we just did the Alamar Blue assay, which is relative to the viability of the cells, and it's the only test that we performed on the cells. So there are not a lot of differences between the three conditions tested.
Okay, great. Barbara?
Yeah, so for our cells, as I shown, the metabolic activity was measured with Alamar Blue assay. We also have a totally different branch of our project where we're really digging into characterization of these cells. I'm not sure if I can really punctually answer this question, but from the assessment of Alamar Blue assay, we observed in this short-term assay that the cells in serum-free were doing very good, sometimes even better than the FBS cells. So,
okay. Great. Another question for both speakers just come in, and they're wondering, have you checked the cell population heterogeneity? And selection of clones? I don't know who wants to take that first.
So for my side, that's why we performed the gene expression analysis. It was really to identify the possible different clones. So for us, it's really possible that the CNTPRA grown cells are expressing a different clone as compared to the two other medium, so the DMEM plus 10% FBS and the ex vivo medium.
Okay.
To answer this question, we did not actually did this classical cell population heterogeneity testing. So yes, I can't really answer it right now.
Okay, no problem. Just to point out that someone in the chat, Thilo Weber, is mentioning that there's going to be a workshop on serum-free media. So there's an email address to get in touch if you're interested. While we wait to see if any more questions come in, I'm wondering, you know, both of these cell lines are connected to, you know, toxicity tests at some, you know, regulatory accepted, some almost or nearly or being integrated into regulatory tests. And I'm just wondering if you have, and Eileen, you mentioned this a little bit in the end of your talk, where you said, you know, when you're choosing your serum-free media, you think ahead of time about the purpose of the test. And so I'm wondering if you can talk a little bit about, you know, if we're going to be using serum-free media grown cells in toxicity testing, what are the things that we should be keeping in mind from a regulatory perspective? Have you thought about that?
For me, for the next step in our lab would be to use really FBS-free medium. Also, we have a co-culture model, and we are working on a co-culture FBS-free medium to grow all the cells and to have our model really FBS-free condition and animal-free condition. And regarding the regulatory point of view, I think we need maybe to set a guideline to help researchers to switch to FBS-free condition. And then it is really important to set a kind of battery of tests to verify that the cells are really answering as they should, but this is really cell type dependent, because for FR ferment cells would be, for example, from my point of view, the presence of surfactant, especially, but then it will depend if you have a, I don't know, a skin cell line or intestinal cell line, would depend on the cell type.
Right, right, great. And Barbara, any thoughts about regulatory?
Yeah, so in our case, this is exactly, as I mentioned in my presentation, why we are doing this, and we are in a better position because we already have tests in place, which is quite rigorously approved. So we now have cells, and we are in the works to actually see how they perform to a known toxicant. So this is this needs to be validated with our results that we have. So what type of differences we might observe or might not is to be discussed after the actual results come in. So then we can actually see what it means for the regulatory purposes. And yeah, so I would say that this is our main goal, and this is really the next thing that we are doing. So
Great. Another question directed at Barbara, wondering about the two animal components still in the culture medium, and if you can say something about, are these essential for cell proliferation and survival? I know you mentioned you're sort of transitioning them out, but yeah, can you say anything about that?
They're not essential for cell proliferation, but as I tried to explain, the biology of the cell is a bit more complex. So sometimes you could just push the proliferation agent, like making cells proliferate. So with animal cells, this is also quite interesting because they could just start to divide and then be smaller in size versus being exactly where you want them to be. So to say that they're essential in order for us to omit them, we would like to replace them with a chemically defined supplement. So we would try to keep it in our mixture because all the supplements that we have are doing something in the serum-free media, which is why they are presented. So
yeah, makes sense. Okay, and I think this might be a very early question, but both of you, if you can say anything about improvements in variability, and I think that means in your results. But yeah, let me know what you think about that.
I think it really is to be seen, and fingers crossed. Yeah, it goes into this direction. Sorry, Eileen.
No, sorry. No, sorry.
So I think we will know more once we actually start have as I said, the results of this testing done.
Great. Eileen?
So for us, we observe, for example, when we expose the cells to SDS. So for the two comparable medium, DMEM plus 10% FBS and ex vivo, we saw a smaller standardization using the ex vivo medium as compared to DMEM, but it's only for this result that we could observe something.
Okay. Last question, do you have advice for determining what features of FBS grown cells are FBS artifacts that we should not want to replicate with replacements? That's an interesting question.
If I may go first, yes, that's really an interesting question because there is also a question, what is the cell line at the primary source of actually what they are supposed to represent and what this long years of culturing in FBS have actually done to the cells. So I can't really pinpoint for some general advice, but I would say that once you start working with a specific cell line, you should really get to know the cell line throughout the history and try to see what makes sense to this particular cell line. So I would go into this direction. This is actually how we have done it. And I was fortunate enough to be in position in such a lab where these cells have been really known, like inside and the outside. So we were able to kind of minimize this questions.
And for human cell lines, maybe what would be nice, and I think it was suggested in a paper from this year, that we can cultivate also the cells in a human serum and compare really the answer to FBS cells, because you have already your test developed using FBS. So of course you want the test working like that, but also you can compare to human serum, as the cells could maybe, it would be maybe more relevant in a human serum situation.
Yeah, that's interesting as well. I know that there have been some discussions at OECD, you know, about the different options, and I think some countries it's harder to get human serum to use than other countries, but I think it's definitely an option for some. It's a good idea. And let me ask this because I don't think either, well, are either of you using any assays with primary cells? And do you have any thoughts about primary cells in serum-free media?
I don't have any experience with that one, so I can't really share my thoughts on this. But if it's a custom-made, custom-built serum-free media that takes into account the cell specificity and also for what it is intended, then of course it would make sense.
And we also do not have really a lot of experience with primary cells, but I think it would be nice also to compare to primary cells because it's really relevant to the in vivo situation.
Okay, so I'm going to go ahead and close out the webinar. We've had lots of great questions and lots of good answers and discussion. Thank you all for joining. And we'll put the recording up on the ASCCT website, and that's ascctalks.org. I think Phoebe just might have put that in the chat. And I will also get that reference that you both used in your talks, the Van der Valk reference, and put that up there as well, so people have a place to go for more information. Congratulations to both of you for your work and your pending publications, and thank you so much for coming to talk with us about it.
Thank you so much for having us.
Thank you.
Thanks, everyone. Have a great afternoon and almost the weekend.